हेलो एवरीवन, वेलकम टू द वेट इनोवेशन क्लास जयपुर। सो आज का टॉपिक होगा स्ट्रक्चर एंड मॉर्फोलॉजी ऑफ बैक्टीरिया इन वेटनरी माइक्रोबायोलॉजी कोर्स। सो द स्ट्रक्चर ऑफ different bacteria can be of different types. Okay, so first we'll talk about the outermost layer of the bacterial cell wall or bacterial cell, that is glycocalyx. Glycocalyx. It is present outermost covering of the cell wall. Next is the cell wall, which is present just beneath the glycocalyx. Next is the plasma membrane. As you can see, this violet color structure is the plasma membrane. This is the cell wall and this is the glycocalyx. Next, this is the cell cytoplasm. This is the nucleoid material or DNA. The main difference of the nuclear material of bacteria to the eukaryotes is that they are not having any nuclear membrane to cover the nuclear material. Next, these are the ribosomes, and ultimately this is the flagella, using which they can move from one place to another in the media. One more structure is there which is not visible here or not mentioned here, is that the plasma membrane of the bacterial cell, they causes the invagination of the plasma membrane, which is termed as the mesosome. Mesosome is nothing but act as a mitochondria, like mitochondrial like function to the eukaryotes. So this is all the cellular structure which is present in the bacterial cell. Now we will talk about one by one. First we will talk about the glycocalyx. So as I told you, glycocalyx is the outermost covering of the bacterial cell, which is present outside of the cell wall. This is formed by gelatin structures, which is of two types. It can be capsule or slime layer. So it can be of two types, that is capsule and slime layer. In capsule, the gelatinous layer will be rigid and arranged proper way. Okay, when gelatinous layer is present in arranged properly and they will form rigid structure, they will call as the capsule. And the similar thing when they will be arranged loosely arranged and diffused then it will be called as slime layer because of the consistency of the gelatinous layer. You can see the example of Actinobacillus. Actinobacillus. So as the Actinobacillus bacteria is having this slime layer outside of their cell wall, so when we see their colonies in the specific media, they look like slimy appearance or sticky appearance. Okay, and to mention about the bacteria which are having the capsular layer outside their cell wall are, we can remember with a mnemonic, that is like even some serious pathogen has nice capsule. Okay, using this mnemonic we can remember all the bacteria which are having capsule outside their cell wall. So starting with E stands for E. coli, S Staphylococcus, then comes to Streptococcus, and then Pseudomonas aeruginosa, then H with Haemophilus influenza, then B stands for Burkholderia, sorry, this P stands for Bacillus, N is for Neisseria, Neisseria gonorrhoeae, which causes gonorrhea in human, and the C stands for Corynebacteria. So these are the list of bacteria which are having capsule outside their cell wall. So what is the use of the glycocalyx or capsule? First thing is they prevent drying of bacteria, as they are hydrophilic in nature. They will prevent the bacterial cell to dry. Next, they are antiphagocytic, they will cover the C3b of phagocytic cell. And prevent the attachment to the bacterial cell. Then the third one is they will prevent attachment of bacteriophages. So the bacteriophages are the viruses which kills the bacteria. So if there is presence of capsule outside the cell wall, they will prevent the attachment of the bacteriophages to the surface of the bacterial cell. So this is all about the glycocalyx. Next, we'll talk about the cell wall. So cell wall is a polysaccharide structure which is composed of different polysaccharides which are linked with the pentaglycine bridge. The polysaccharides are N-acetyl glucosamine and N-acetyl muramic acid. These polysaccharides, they are linked with each other with the amino acids and pentaglycine bridge, and they will form a cross-link structure which is known as the cell wall. So the cell wall, they will be responsible for 10 to 40% of the bacterial weight. So the structure will be N-acetyl glucosamine, which is linked with beta 1,6 glycosidic bond to N-acetyl muramic acid with some amino acid same with here is L-alanine, glycine, glucosamine, alanine. They will be present with pentaglycine bridge. This is the structure of cell wall which is made up of peptidoglycan and this is the structure of the peptidoglycan. Now we will see the cell wall of gram positive and gram negative bacteria. In gram positive bacteria, the cell wall is having three parts. That is, first one is the cytoplasmic membrane, then there will be periplasmic space, and third there will be peptidoglycan layer. This is the peptidoglycan layer, this is the periplasmic space, and it is the cytoplasmic membrane. So together they will form the cell wall of the gram positive bacteria. In gram negative bacteria, they will be having this cytoplasmic membrane followed by periplasmic space, then they will have a loosely attached peptidoglycan layer, and finally they will have outer membrane, which is absent in gram positive bacteria. So as in gram positive bacteria they doesn't have outer membrane, so when they will be treated with lysozyme, they will form a structure called protoplast. They will form the structure called protoplast with only cytoplasmic membrane or plasma membrane. In case of gram-negative bacteria, they are having outer membrane apart from the presence of peptidoglycan layer, and there will be presence of lipopolysaccharide LPS, which will produce endotoxin, or which acts as a endotoxin and causes endotoxin shock to the host organism. Okay, so when we'll treat the gram-negative bacteria with lysozyme, there will be less impact of the degradation of the cell wall in the gram-negative bacteria, and they will form spheroplast structure with presence of outer membrane. So now if we see what are the uses of cell wall. First of all, due to the presence of cell wall in the bacteria, the definitive cell strip is present. Due to the presence of the cell wall only, there are different shape of bacteria occurs in the environment. Then next is they will prevent osmotic lysis, means they will prevent or they will maintain, ये क्या करते हैं, osmotic balance को maintain करते हैं inside and outside of the cell, means cell के inside osmotic pressure and the outside of the cell the osmotic pressure में balance बनाए रखते हैं, इसमें इसका योगदान है. And the third one is prevent phagocytosis. The fourth one, we can say, due to the presence of cell wall only, we can identify different bacteria using differential staining. So this is all about the cell wall of the bacteria. Next, we'll talk about the cytoplasmic membrane. Cytoplasmic membrane is formed by phospholipid bilayer with intermembrane proteins. These are phospholipids. These are intermembrane proteins. And these structures are porine proteins, which will help in the transport of different substance across the cell membrane. So this cytoplasmic membrane structure is called as the fluid mosaic model. And what is the main use of the cytoplasmic membrane? That is the selective transport of essential nutrients across the essential nutrients across the cell. Okay, next is the mesosome, which is present in some bacteria. It is formed when bacterial cell membrane invaginates towards inside of the cell cytoplasm. So this is formed. So this mesosome acts as the mitochondrial equivalent organelle. Just like mitochondria acts in the eukaryotes, this mesosome helps in the bacteria in case of cell respiration and to produce energy. Next comes to the bacterial motile organs or the bacterial cell structure which helps the bacteria to move from one place to another or gives the motility to the bacteria. So this I'll talk about the bacterial flagellar structure. This is the structure of bacterial flagella. This is the filament. This is the hook with which it joins with the basal body. So this is the basal body, this is the hook, and this is the filament. This three part is present in the single bacterial flagella. So in here we need to remember one thing, that in basal body there is a flagellar motor इसमें एक बैक्टीरियल फ्लैजेलर मोटर होता है जिसके वजह से द फिलामेंट मूव्स फ्रॉम वन साइड टू अनदर, एंड need to remember that this flagellar motor is the strongest protein this flagellar motor. They produce the strongest protein motive force in nature. As we can see, the structure of flagella is very small, but they are having this flagellar motor which is very strong in nature, and they will produce strong protein motive force, which because of which the filament moves and the bacteria can move from one place to another in the while growing in the bacterial cell media. So based on the arrangement of the bacterial flagella, they are of different types. First is atrichous bacteria, where there is no presence of any flagella, like Staphylococcus, Streptococcus, Bacillus, Clostridium, all are the examples of atrichous bacteria. Then monotrichous bacteria, where there is presence of single flagella at one end, that is like Vibrio. When there will be presence of one flagella at the one end of bacteria, it will be called as the monotrichous bacteria, and the example is Vibrio. Then amphitrichous is presence of bacterial flagella at the both end of the bacterial cell. Single bacterial flagella present in the both end of the bacterial cell. The example is alkaligens. Alkaligens faecalis. Next is the Lophotrichus. In Lophotrichus, there will be tuft of flagella or more than 2 or 3 flagella occurring at the one single end of the bacteria, that are causes Lophotrichus, that will be called as Lophotrichus bacteria. Example are Pseudomonas, Putida. Pseudomonas putida. Then comes the fifth one, Cephalotrichus. Cephalotrichus bacteria are those bacteria where bacterial tuft of flagella or more than 2 or 3 flagella is occurring at the both end. दोनों तरफ बैक्टीरिया सेल के दोनों तरफ tuft of flagella देखा जाता है. So those are Cephalotrichus bacteria like Pseudomonas, aeruginosa. Pseudomonas aeruginosa. Then finally the Peritrichous bacteria. Peritrichous bacteria are those bacteria जिसके चारों तरफ flagella पाया जाता है. Means the flagella is present all the sides of the bacterial cell. Example are E. coli, salmonella. Okay, so this is all about the flagellar structure of the bacteria and the different kinds of bacteria due on the presence of bacterial flagella in different position. Next, we'll talk about the filament. Oh, sorry. The fimri. Fimri is also like flagella. They also help in the attachment of the bacteria to the other bacteria, and they are also called as the pilus. When they are present as a single fimri, they will be called as the pilus, and as a group of fimri, they will call as the fimri. So when they will occur as a singular, they will be called as a pilus, and in plural, they are termed as fimri. What they will do? They will help in the attachment of the bacteria to the other bacteria. Attachment of one bacteria to other, and the main important is the conjugation. So conjugation is the process where one bacteria to another bacteria, the genetic materials are transferred. एक बैक्टीरिया से दूसरे बैक्टीरिया में जेनेटिक मटेरियल ट्रांसफर होते हैं, एंड वो कैसे होते हैं? जब एक बैक्टीरिया के बाहर डिफरेंट वेरियस फिमरी होते हैं या पाइलस होते हैं, वो दूसरे बैक्टीरिया के साथ अटैच होके ब्रिज बनाते हैं. Through this bridge, they will transfer genetic material, that is DNA, from one bacteria to the other. So this is all about the fimri. Next, we are going to talk about the bacterial spores. This is the structure of the bacterial spore. This bacterial spore is present in some bacteria, which will help the bacteria to withstand harmful environment. When the bacteria, जब bacteria कोई environment में रहता है, like when bacteria gets inside our body and our body cells will attack those bacteria. So what they will do? They will germinate spores. They will germinate these spores, and this will help them to resist the phagocytosis, which will be by the our body cells will cause the phagocytosis. So they will prevent, these bacterial spores will prevent the phagocytosis. So the bacterial spores are having the core well, which is mainly formed by calcium dipicolinic acid, surrounded by the membranal structure. This bilayered structure, and the core well with calcium dipicolinic acid, these are the main component which producing antiphagocytic property. Okay, so this is all about the structure of the bacterial spores. Now the arrangement of the spores in bacteria, depending on the various position they are present in the bacterial cell, they are classified in three. They are mainly classified in three types. There, first is when the spore is centrally placed. In bacterial cell, when the spore is present at the center, it is termed, those are example Bacillus cereus, and Clostridium botulinum. In those organisms, the spores are present in the center of the bacterial cell, which will cause the bulging of the bacterial cell. Okay. Next is the terminal. The example are Clostridium tetani. Then the third one is when the spores are present in the subterminal region. Where we can see this in Bacillus subtilis.